Good morning, students. Good morning, everyone. In 2 minutes, the section will be start. YouTube Live and Facebook Live is also going on. Please check it there also. For facing trouble in airmit, you can join in YouTube Live and Facebook Live also. స్టూడెంట్స్ మీకు ఏమైనా డౌట్ ఉంటే సార్ అడిగినప్పుడు చాట్ బాక్స్ లో డిస్ప్లే చేయండి అమ్మా. మీ డౌట్స్ తెలియకపోతే ఎవరికి మీరు సార్ గుడ్ మార్నింగ్ సార్. హలో. స్టూడెంట్స్ మీకు ఏమైనా డౌట్స్ ఉంటే చాట్ బాక్స్ లో రెస్పాండ్ ఇవ్వండి అమ్మా. ఎందుకంటే సార్ మీకు డౌట్స్ క్లారిఫై చేస్తారు. మీరు ఎవరు మిస్ అవ్వద్దు. మెయిన్లీ మీ డౌట్స్ ని క్లారిఫై చేయడానికే మేము ఈ ప్లాట్ఫామ్ ని తీసుకొచ్చాము. మీ ఏ డౌట్ అయినా పర్లేదు చాట్ బాక్స్ లో పెట్టండి. ఐ విల్ రీచ్ టు సార్ అండ్ ఐ విల్ క్లారిఫై యువర్ డౌట్స్ డెఫినెట్లీ. ఓకే. జస్ట్ 1 minute లో సార్ జాయిన్ అవుతారు. జస్ట్ ఐ హావ్ కాల్ టు సార్. ఎవరు వెళ్ళొద్దు, లెఫ్ట్ అవ్వద్దు ప్లీజ్. సారీ ఫర్ ద డిలే ఆల్సో. Good morning, students. Good morning, everyone. We are having YouTube Live and Facebook Live also. Please check it. You can join there also. Please don't left anyone. Yes, now Sar is going to join. Sar has started to join the section. Please don't left from the section. It's requesting. And once again I'm saying really sorry for the delay. Good morning students, good morning everyone. Sorry for the delay really. I'm saying sorry to you once again. Sorry just now to join. Please don't let anyone. This is requesting from my side and from our organization. Please don't leave the section. Please. It is very important for you. That's why I have something this much for you. Please. We are there for you. Sorry, once again, sorry. Please don't leave anybody the section. The section will be start now.
Hello. Can you hear my hello?
Yes, sir, I can hear your voice. You can.
Let me share my slides and just confirm if it is done. We are ready to start.
Yeah, okay. Yeah, it is visible. You can continue, sir.
All right. So, very good morning to all of you, and uh, welcome back to Drug Discovery Development course, ongoing online course, an initiative by Andhra Pradesh State Skill Development Corporation. So probably today we'll try and finish the remaining topics. So let's get started. We are already late by a few minutes. My apologies for slight delay in logging in. So let's start. Couple of very important topics. We'll start with discovery chemistry. So, discovery chemistry basically is a part of chemistry. I mean, it's chemistry only, but just within the chemistry, discovery chemistry relates to the research of new drugs. Okay, where chemistry alone doesn't really work, but it works in tandem with other disciplines like biology and physiology. So keeping all this integrated work, whatever chemistry is applied, that's called discovery chemistry. Okay. So the drug discovery development, whatever we have talked about, as you might have understood by now, that it is driven mainly at initial phase by knowledge of chemistry of the molecules, because the whole research, whatever testing you do, biology, biological testing, toxicology, or anything in vitro, in vivo, but everything is based on a novel chemical entity, right? So it is completely driven by the knowledge of chemistry of the molecules and its association with the life processes. That is, of course, because it has to act on some or the other target in your body. So that's why we say knowledge of chemistry drives drug discovery and development. So what is the role basically of chemistry? So you see, not only in designing the drug, but if you carefully understand the physiological processes within our body, even that is as a result of a chemical reaction, one or the other chemical reaction at a molecular or a cellular level, right? So the natural physiological process is distributed. Whatever natural physiological process is there in our body, whenever there is any intervention or when it gets disturbed, the state of body is in a disease state, correct? So whatever physiological activities, processes are running inside our body, if there is any disruption in that, will lead to a disease state. We all agree with that, because even for diabetes, like there are all mechanism. I'm just taking an example of diabetes, that once you take a drug, it gets digested, glucose is released in the blood. Now there is an organ called pancreas, from where the insulin gets released, and it balances that released glucose in the blood. Now, the moment there is any impact on the pancreas, it doesn't really releases insulin, or the required amount of insulin, or there is a delayed release of insulin, or it is acting slightly slowly. The immediately your blood glucose level will rise, right? And the disease state would be diabetes. If it is a prolonged thing, then it will be a diabetes. So whenever you take food, whatever glucose is produced and then it is released in the blood, it cannot be counteracted. It cannot be utilized by the itself. So that exaggerated level then becomes a disease state called diabetes. So that's why we say that normal, natural, physiological processes, as long as they are working normally, it's fine. Any disturbance in that, then there comes a disease state. So drugs or medicine basically are chemical substances, right? And we use this to treat, cure, prevent, diagnose a disease, eventually to ensure the well-being of a person, right? Because a person is free from disease, then that is what we call a state of well-being. So medicinal chemist basically discovers the drug or identify a chemical compound. Of course, there are different means and ways how we can do it. It can be completely novel, or it can be a slight modification of existing structures. It could have been derived from some natural compound, but Basically, chemist really identifies the chemical compound which really can cure. So many a times we say that this particular plant extract or this particular herbal extract, it has antibacterial activity, it has antiviral activity. So what are we trying to do? We are trying to uh, because based on the structure and the existing literature, we know that this can really bring about this effect. So for herbals and all, of course, we do a reverse pharmacology. We don't know the exact structure or exact active pharmaceutical ingredient or what is an active ingredient in that particular extract which is responsible for this, because it could be a mixture of few active ingredients within a herbal extract. So that's a little challenge, but for a novel chemically synthesized compound, it's very specific. So it could be one particular chemical structure chemists would have come up with, which can be highly potent, which is very safe, and then you work around the analogs or work around the small modification of the structure, and you can make it a drug candidate. Right. So the medicinal chemist discovers the drug or identify the chemical compound to cure the disease with minimal side effect. Right. Because the whole aim of having of doing drug discovery development is to come up with novel, safe, and efficacious. Of course, if I remove novel, to come up with safe and efficacious drug. Okay, so he or she, which is a chemist, will design and synthesize novel compounds having pharmacological activity. That would be the first step. Now, the chemist is also responsible for developing the manufacturing process, because in a research scenario, when you're doing the testing in preclinical or early discovery, you are going to utilize very small amount of drug in mgs, but finally when it makes it as a drug candidate, then you need the same compound in a larger quantity in grams. Finally, when it is going to be launched, you may need it in kgs, right? So chemists will develop the manufacturing process also, and formulations also whenever required. They and produce it in plants. They do analytical studies. So actually chemistry role is everywhere, not only in designing the molecule, in manufacturing, sometimes in formulation development, and analytical chemistry is anyway a very important stream which really helps in early phases of discovery and through different phases, because analytical chemistry will be applied. Okay, engaged in discovery chemistry should have a knowledge of, this is very important, of other fields. I always say this based on my own personal experience also, that say you are not a good chemist if you don't understand biology, if you are into discovery research. And same way for biologist. A biologist is not a good biologist if he doesn't understand chemistry, right? Because they are interrelated. Same way, knowledge of all the other allied fields. Biology includes everything: biotechnology, microbiology, pharmacology. So that's how we should always try and learn. Because I'll give you an example, a real life example from drug discovery development. Suppose if you are a chemist, and if you don't understand the ADME properties, basics of those properties, you will never know the liabilities may have, right? So you cannot modify your structure accordingly. Same way, if you're a pharmacology scientist and efficacy study scientist who is doing the biology in vitro or in vivo to establish the efficacy of a compound, don't understand chemistry, then actually you will never be able to suggest anything to a chemist that for a good efficacy, maybe this small structural change can help, right? Any suggestions, except for sharing the data back with him, you won't be able to share anything else or any suggestions. So that's how I feel. It's vice versa. A biologist should also understand basics of chemistry, and chemists should understand basics of biology. So new drug discovery research, I think we have understood now with this many classes, right? That some idea in your mind for a novel drug, we plan whether it has like properties, whether patentability is there or not, whether this target is approachable or not, and then you do all kind of early target identification, validation, hits, lead, all that discovery in a high throughput fashion. We try to do it. And then at some point in stage, we will go to animal testing and do the preclinical development. Then finally, we go to clinical trials, and then all those things are completed, phase 1, phase 2, 3, then it registered, and then you get a new compound. So this is the typical drug discovery development path for any novel drug discovery development research. So let's spend few time on choosing a disease. Just to give you an example, that see, pharmaceutical companies, when they choose a disease, there is definitely a commercial enterprise. I mean, a commercial interest, and it is true because I'll give you a current example. Say, for example, COVID scenario is there. Of course, to combat this disease is the major goal. But at the same time, small or maybe on larger percentage is driven by commercial interest also, right? If companies are coming up with vaccine, the only goal is not social service, right? They want to combat COVID. We want to prevent COVID by having a vaccine. But each of these companies, they have a commercial interest too, right? They're going to earn money out of it. Maybe they may earn less profit and try to make it for more benefit of the people, but eventual goal is commercial. So pharmaceutical companies, see, when COVID was not there, did any of these companies even think of developing such vaccine? They may be working on other vaccines which for the prevailing disease, but now many companies are working on COVID because they want to take some commercial advantage out of it, right? So pharmaceutical companies, they always have a commercial interest. So many a times it's not that straightforward exercise of choosing a unmet medical need or a disease. What happens is it is a fact. It is not fair, but it is a fact. That's why I'm sharing it with you, that many a time companies will avoid product that would be consumed by individual of lower economic status. Okay, why? Because they can't afford the drug, so companies may not select those disease, right? Majorly, see why in western countries lot of drug research is going on obesity. Why? Because it is like considered the first world countries, right? So obesity, like the people would be able to afford those drugs. So a lot of research efforts are going on to come up with anti-obesity drug, right? Cardiovascular or even cancer. The affordability is also one of the factor which is considered by pharmaceutical companies when they decide to choose a disease. From research perspective, then you can select any disease, right? But whenever it is done by a pharmaceutical company, the end result, because eventually, as you have understood, the whole process is very costly. A lot of cost investment is happening. So eventually, we want a return on investment. So that's the reason all these factors are kept in mind while deciding the disease. So if you see an R&D expenditure, majorly 30% expenditure happens where? In clinical. In target identification, validation, that is also one of the major aspects. In total, around 30 to 40% investment happens there, right? And then preclinical and optimization together around 20 to 25%. So, and 30% up to in clinical trials. So, whatever $2 billion of investment we have talked about in drug discovery, this is the breakup of that. Around 30% in clinical, around 25% in preclinical and optimization, and around 20 to 30% goes in target identification and validation. So this is a percentage wise, same thing what I have shared within the clinical trials. For 1, 2, 3 all together around 30% they cost, and phase 4, which is post marketing surveillance, cost you around 10% or 11% of expenditure, right? If you talk of drug discovery approach, then it can be a de novo approach or me too approach, right? As the name suggests, it can be completely novel or it could be a me too approach. So what happens is, if you compare the expense versus return, okay, then in me too the percentage expense is 20%. In de novo, as you may understand, me too is like you're trying to copy something which is existing, right? So the expense is definitely less. When you're trying to do something new, so naturally there are a lot of challenges, as we have seen: the target identification, validation, screening, early drug discovery, exploratory studies, preclinical development, in vivo testing. and then clinical trials and all. So the investment is more. So de novo, it is 80% expensive. But when you talk of return, me too, you have to 50%, right? And it is almost same. So investment is more in de novo, but return when you talk of, they're almost same. Either it is me too or de novo approach of drug discovery. Let's take an example of statin. So you see lovastatin, pravastatin, simvastatin, rosuvastatin, all this structurally, I would say not exactly analogs, but yes, they have come from a similar structure, right? Now you see, lovastatin 1987, it was launched, point 0.2 billion US dollars of sales it made actually. Simvastatin 2 years after, $2 billion. You see, in same way pravastatin, all from different companies, but all of them were really a successful drug, except for servivastatin, uh, because that was withdrawn. Okay, and in this is the basically there are statins, right? So they are used for cholesterol, or in other words, we say for LDL reduction or to increase the high lipids, right? High density lipids, and to reduce the triglycerides in the blood. So that is the role of all of these agents. So these are like similar compounds, but you see in the returns are fantastic, right? Let's take an example of anti-ulcer, right? Omeprazole, correct? Lansoprazole, pantoprazole. These are all the structural modifications you see their year of launch, but all of them made huge business, right? So return is really huge. So in past, drugs were discovered. Definitely, we have talked about it like serendipitous approach, like serendipitous discovery. That is discovery by chance. But now, the major thing for us to understand the difference, as you know, me too is the existing one. So the collection of structures and the data of known compounds for a particular target, correct? Whereas here you have to identify a target and then you have to validate, right? And then you have to go for proof of concept study to validate your target. Then you do a get a hit for the hit screening, then you get a lead, and then optimization, and then finally optimization of a compound to improve the drug-like properties. Whereas here you have the collection of structures or from the known compounds, and then novel compounds you can design from that, or using computer aided drug designing, or once it is done, then the process is same. You have to identify the lead, and then you optimize the lead, and then you improve the properties. The initial, there is a difference between de novo versus me too. Remaining is pretty much the same. In de novo approach, we have talked in detail about this. Let's jump deep dive into it. Say target selection could be cellular, so role of biotechnology. So we'll go into each of this and see what exactly is you mean by knowledge of genomics or proteomics or bioinformatics. So target selection, if you start with, so an agent with particular biological action, as I already mentioned, by and large, this is going to be a molecular target. Okay, so it could be a pathway, it could be an enzyme, it could be some protein interaction. So any of this molecule, or maybe what do you call some receptors. So these are all the targets by and large for most of the novel drug targets. So this is example taken from Nature Reviews. If you see for the molecular targets, G protein coupled receptors, around 45% targets are based on receptors, targeting the specific receptors. Around 28% is for enzymes. So receptors are majority of the targets. Then comes the enzymes, hormones, hormonal factors, nucleic acids, ion channels, or nuclear receptors. So by and large, this is the distribution of molecular targets in drug discovery. Right. This is specific examples are given that within receptors, G protein coupled receptors, ligand gated ion channels, receptor tyrosine kinases, in enzymes, oxidoreductase, transferases, hydrolases. So this is detailed in a Nature Review article. They have given a wonderful article on this. If you get time, you should read that. That this targets distribution, and in each of the target, what are the major targets and which are the disease they really target. They really intervene with. That is completely defined. Same way for the transporter proteins, and other interactions or protein interactions. So majorly anti-inflammatory or hypertensive or anti-neoplastic or anti-allergic or immunomodulation. So these are all the disease areas, therapeutic actions which can be elicited by acting on this molecular targets, right? So the when you say cellular and genetic targets, so it starts with identification of function of a potential therapeutic drug target, and then its role in a disease progress. That by what is it, the inhibition, is it the induction, or is it the interaction? What particular function or the process disruption will lead to a disease state? That is what you need to understand. If it is enzyme, same thing, it's upregulation or a downregulation, or signaling pathways, is it blocking that pathway really creates a disease, or opening that pathway creates a disease. So those are the functional process which you need to understand about these molecular targets in order to identify the target, correct? So there are several techniques people try and do. They may select a group of targets and then they narrow down on one target, or they can start with one target and then move around that entire group of targets. Okay. So basically, genomics is also equally important because, as you know, study of genes and their function, and then what is the aim to structure of the genome. As you know, human genome is now fully understood, right? So basically, if you understand the structure of the genome, the mapping of the genes, sequencing of the DNA, that will really help you to really further narrow down your cellular or a genetic target for the disease. Okay. So whenever you say this is just like in human genome, it consists of, as you all know, ACGT bases, the nucleotides. So more than 3 billion nucleotides are there. So more than 35 to 50,000 genes are being encoded. So imagine how many potential, how much of a potential you have to have a disease target, right? 35 to 50,000 genes are encoded. So huge potential to decide on the molecular targets, right? So Drew's estimate, if you take then the number of genes implicated in disease, both due to the defects in single gene or those arising from combination of gene itself, is about 1,000. So based on 1 to 5 or 5 to 10 linked protein per gene, basically the potential drug target would vary somewhere between 1,000 to 5,000 to 10,000 molecular targets at a gene level, right? So nowadays even SNP libraries are available. So single nucleotide polymorphism, these libraries are available for students, I mean for the people to decide from. Uh, my I think some disturbance is coming from this. Can you just mute? Yeah, thank you. So, so SNP libraries are actually available for chemists or for a biotechnology scientist to choose disease targets from. Okay. In terms of proteomics, study of proteome. So the complete set of proteins, as you know, produced by species using a large technologies of scale protein separation identification. That's the whole process. So you know, there's a lot of complex biological systems uh, basically in our body lies, and the basic unit, if I had to select, that would be a protein. So as in today's scenario, when we say biologics, so basically we're talking of proteins, right? At the end of the day, anything or an enzyme or whatever you say, they are proteins, right? So protein level, that disease processes become, it's like 91% of the drugs would act on one or the other kind of your proteins in your body, right? So therefore we say that analysis of these proteins, which includes protein or protein nucleic acid or protein ligand interactions, will really help you in understanding the molecular target, right? So please remember, any protein or protein ligand or protein nucleic acid interactions, and then again there are tools, there are softwares available through which you can actually understand these interactions also once you decide a particular disease target. Okay. So as I mentioned, this can be done in a high throughput fashion. Okay. So nowadays, protein scale up or separation, identification, characterizations can be actually done in a high throughput fashion. So basically, target identification with proteomics, if you want to do, then you have to compare the protein expression in a normal tissues versus a disease tissue. So that's one way of doing it. Like a normal tissue, you can see the protein expression, and in a disease state, if it is an organ specific and there's any change in expression, then that becomes one of the target for you to decide for a disease treatment. Okay. And there are different technologies, maybe mass spec nowadays, or electrophoresis, gel electrophoresis, 2D page. These are all the techniques you can use to separate the proteins. Bioinformatics, again, very important, because bioinformatics is extensively used, because basically it's nothing but it is analysis of biological data. So there are, that is again big data. I mean, is analysis as also nowadays also very important, and then to enhance our biological research, bioinformatics knowledge is very important. And it can help, your knowledge in bioinformatics can help you at not only target identification, but during the screening of molecules using softwares also bioinformatics helps. And pharmacogenomics also, with the data set available in the at in form of softwares, can really help you. So bioinformatics also really help to identify a particular target apart from proteomics and genomics. Okay. So they actually, if I talk of very specific sequences, so what it does is they convert or transform a raw sequence into a meaningful information. Okay. So what is a raw sequence? It can be a gene, any random sequence of a gene, and they're encoded proteins. So this can really, when you compare it to the whole genome, so it's very easy where the difference you have seen between a disease and a normal tissue or a normal individual. So if you compare that comparison, earlier it was really tough, but now with advancement in bioinformatics, it can be done easily, sitting on your computer using those softwares, right? So that's the reason bioinformatics knowledge is also very important. So as I said, genome informatics, target selection, molecular modeling, dynamics, modeling, all this comes within the purview of bioinformatics. Even chemoinformatics, that is all related to chemical. So the structure based, okay, QSAR, structure activity relationship, the molecular diversity, ligand based drug designing, all that comes within the purview of chemoinformatics. So bioinformatics as well as chemoinformatics. So today is a scenario of technology development and its application. This is something new, a decade ago or a couple of decades ago. In drug discovery and development, this was not there, but now all these tools are available to help you in doing drug discovery in initial stages. Okay, so screening to generate hits. We have covered target identification, role of genomics, proteomics, bioinformatics. In earlier classes, we already talked about the lead optimization, all the in vitro assays and all. Now screening to generate hits. So lead optimization, prior to that step, is to identify a lead, right? To generate a hit. So how can you do it? By doing a functional assay or a binding assay, right? So compound binds to the cell surface or a receptor, and if it can be measured, that's called a binding assay. So how strong a binding is, if that binding can be measured, that can also give you which particular target, I mean which particular structure is able to bind to the receptor, right? If you can measure that, that becomes a binding assay. Suppose if it can evoke a cellular response, okay, and if you can measure that response, that becomes your functional assay, right? So either it can be a cell based or an enzyme based, but by doing this assays, you can really generate screen your molecules to identify the hits. Nowadays, it is done in a high throughput fashion, millions of compounds can be done. Gone are the days when they were done individually using small append-off tubes. But now in 96 well plate to 384 well plate and even 1536 well plates are also available, and using liquid handling units in an automated fashion, so you can do this hit generation in a real high throughput fashion. Okay, and of course knowledge base, as we have talked about, computer modelings can be done, right? Structural knowledge, chemoinformatics, all this thing, bioinformatics can be used to really narrow down your screening process and to generate hit in a smallest possible time. So HTS plays an important role, high throughput screening, right? Identification of one or more positive candidate extracted from pool of possible candidate based on the specific criteria. So whatever binding assays or a functional assay or whatever assay you want to do, if you can do it in a high throughput fashion, you are actually doing it in a quicker fashion, right? So there are chances of getting the hits out of it would be more, and it can be done in an automated fashion, so people involvement is also less, so it can be real quick as automated HTS. So if you have to define, when you screen, I mean when large number of compounds are rapidly tested for their ability to modify the properties of selected biological target, so this can be applied in high throughput screening. But nowadays high throughput screening is applied everywhere in all the analytical tools, HPLC or a mass spec or a spectrophotometer or whatever tools you are using, you can do it high throughput, right, fashion. When you do this high throughput screening initially, our purpose is basically you want to test the ability to modify the properties of a selected biological target. So goal, what is the goal? Our only goal is to identify a hit or a lead. Okay. So the simplest target centered approach. So you used a very specific target based assay. Okay. And a robotic automation so that thousands of molecules in just few days of time can be run. So first, there are two critical aspects to it. If you want to do it in such a high throughput fashion, your screening to identify a hit or lead by doing a particular target based assay, first foremost thing is you should have a library of compound. It should be available. You can't just search your compounds and daily you can do. You should have a libraries of compounds already available with you. And any one simple assay should be identified or developed. Only then it can be done in a high throughput fashion, right? So the simpler the assay, the automation of that would be very simple. Say for example, some enzyme or some biological matrix has to be pipetted out, add your compound, mix it up, incubate, quantify, something like that. It's just an example. So very simple assay has to be in place, huge libraries of compound should be in place, then you can do this high throughput screening in a very simpler manner. This is just few example pictures I have seen, like robotic arms in such a large scale it can happen, and you see it is 1534 well plate. So this is the extent of high throughput, and I'm fortunate enough to have visited a couple of such labs in my career where things were really happening high throughput. This is even better, but I have been to labs where they do screen by screening assays for at least hundreds of compound within overnight. Okay, you can program and automated way it will be done. Next day when you come to the lab, you'll have data from hundreds of molecules ready for you to analyze. So this is how a high throughput screening is done nowadays, right? As I mentioned, in terms of detection method, in instrumentation also high throughput can be done, be it spectrometry, mass spectrometry, chromatography, X-ray diffraction, everything can be done in a high throughput fashion. Even in spectroscopy, if you see fluorescence or internal refraction or NMR or FTIR, you can actually do high throughput fashion. In chromatography, as you know, GC, HPLC, mass spec, reverse phase, ion exchange, hydrophobic interaction, basically all of this can be done in high throughput fashion. Either they have an auto sampler where you can run in a 96 well plate or a 384 well plate format, or they are sample manager additionally available where there are racks available where you can keep 96 or 384 such 10 plates, and that robotic arms job is to you, of course you have to program it with your system. It can just take the sample, well, I mean the plates, and then put it into an auto sampler. Once it is over, it will take it out, it will put the another one. So those things can be done, or samples can directly be taken from that. There are so many technologies available. So high throughput can definitely be done in a chromatography as well. Again, in ITS, in calorimetry, in microscopy, atomic force microscopy, confocal, all this thing can be done, or a microarray technique. All that thing can be done in a high throughput fashion. So, as I said, the heart of the any HTS system is the plate, either 96, 24, 96, 384 or 1584 well plate. So the tray, which consists of tiny wells, okay, where actually the assay reagents and the samples are deposited, and whatever reaction happens, that is monitored, and once you measure that, you will get a result in whichever way, inhibition or induction or quantification. So basically a well plate with a well, where you can add a reagent, chemical and a samples, and you can observe the reaction, right? Configuration, as I mentioned, 96, 384 or 1536. And it is typically, I'm sure most of you have seen a plate, but just to mention, this is how a plate looks like. You will have a well, and then that's where you can do your reaction. So the reagent is coated on the well. Yes, nowadays, even for protein precipitation, if I want to do just an example, already pre-coated wells are there. So already there is a coating available, right? So reagent is coated on the well, added in the, or it is added in a liquid form to in a simplest well. You just can add the what do you call samples, and plate and wells is incubated, signals are given out, and then finally whatever reaction of that reagent and the sample has happened, that has to be studied, detection, wire detection. Okay, so that is basically called as well place. If you can, I'll just take one moment, I'll show it to you how it looks like. All right, so I'm glad that this is already available. So I could show you that this is how, if you can see my screen, then this is how it looks like. Let me just minimize that. And if you can see my screen, I mean, if you can see my video, then
Yeah, video we can
yeah,
see your 1 minute, sir.
yeah,
Can you stop the sharing? Yes,
yes.
right?
So this is how basically a typical well plate also looks like, and I have typically taken this because it is pre-coated. I mean to say there is a filter on the back side. If you see some whitish material you are seeing, but it is basically inside the well. So basically, this is meant for protein precipitation. It's a 96 well plate, and it comes up. It comes with a cover also. Okay, so you can put this. So this is just an example how a 96 well plate looks like, and most of the assays, either cellular assays or sample processing assays or a biological assays or a binding assays, whatever I'm doing can happen in this. So there are 8 wells, right? 12 wells. This is how we use it, and nowadays they are pre-coated. So if I have to show you inside, they are already pre-coated ones also available. Okay, if it is not pre-coated, it will be very transparent, so you can see just like this, it will be transparent, although it's a cover, but otherwise the 96 well plate also looks like this. So this is the heart of any high throughput screening, either in an initial stage or for going further. Even let me tell you, when you do in vivo testing, whatever blood samples you're getting, even those samples also can be processed, and generally are nowadays processed using this well plate format only. Gone are the days when you were using test tubes and those things, because that was not a high throughput fashion, but now everything can be done just like this, using a 96 well plate format. Okay, so let me go back to my presentation. I'm sharing. Yes. Are you able to see my screen, Maheshwari?
Yes, sir. I can. You can continue.
Great. Yeah. So that was a live example of how it looks like, and cell-based assays refer. Okay, so now we moved on to the next one. Cell-based assay basically. Already we have talked about hepatocytes, enterocytes, but this is from a perspective of a biological assay when you're trying to screen your molecules. So cell-based assays, any of a number of different experiments based on a live cell can qualify as a cell-based, either a metabolism or a binding assay or a permeability assay. Anything where you use a live cell are called as cell-based assay. Okay, they are important, right? Because this can be compared with other in vitro assays. If you do any chemical assay or a biochemical assay, cell based are more important because somehow they are more relevant to the actual life system, because they can express some enzymes, they can express some proteins which are very specific available in the live system. So cell-based models have more significance compared to non-cell-based models. Okay, where fundamental unit of expression is cell, of course, and in either cell population or a single cell. Nowadays, even single cell assays people do it. The enzyme-based and the cell-based assay system consists of, as I mentioned, receptors or mimetics of receptors. It can be expressed on this. So please remember, any cell-based assay will have four basic things: a cellular component, right? A cell line or a primary cell line has to be there, or a primary cell culture or a secondary cell culture. You need a substrate, the molecule that record the cellular response, and instrument to conduct or to monitor the entire assay, and an informatics component to analyze the data, right? So very simple. You need a cell, you need your substrate, your compound which you want to test. Either you are testing the efficacy or you're testing the toxicity, or simply you are checking the metabolism. You need a substrate, you need a cell line, and there should be an instrument where you can see this impact in terms of inhibition, induction, or permeability or quantification. You should be able to do using some instrument, and then finally whatever data you get, you should be able to interpret that data or analyze that data, right? So advantage of cell based over biochemical, as I mentioned, analyze the sample compound activity in an environment that is similar to the live system. Okay, so don't want to re-emphasize again. Many cell lines are very common cell lines which are generally used, like human cell lines, DU145, PC3, MCF, MDA, T47. So there are different cell lines which are generally used for biological, as a biological assay. Again, I don't want to spend much time on this. This is a very typical example of one of the like before you do a fax assay, how exactly it can be done in a cell-based model, right? So by doing this cell-based or a non-cell-based enzyme assays, you will come up with your heat identification. And from this, once you have your heat, then actually, basically, when you select the heat, you concentrate on two aspects. Majorly, it is the potency. Okay, because you have so many compounds to screen, the only criteria which can give you the hit initially, right now you're not looking for any safety liabilities and all those things. All you look for is potency. So you define your acceptance criteria, then any compound which can show above this much of potency, or potency at a lowest concentration, are my hits, then you can screen your compound. So what is the criteria? Potency, correct, and selectivity. Of course, selectivity, because you don't want off target hits. So we want it should be highly selective and it should be highly potent. So once you have, so in lead discovery, basically small molecule modulators of a protein function. Okay, so you have to practically, as I already mentioned, these are the plates which I have just typically shown you. So you have to actually synthesize such molecules once you have the lead. Okay, and then we run it for the potency assay and combinatorial chemistry, because after you do the potency testing, you suppose if potency needs to be increased, Then you go back to the chemist and you request them that please modify your structure. Can we further increase the potency? So all that thing can happen in a high throughput fashion. So that is called lead optimization. And lead optimization, again, so many things. Let me just decode it for you. So this is a liquid handling unit. If you can see my cursor, where you can do it in a high throughput fashion. This is also from a Tomtech, right? This is how it looks like. These are all high throughput fashion. You can do it actually. So just wanted to give you an example. So the lead compound is a structure that has come activity against the chosen target, but not yet good enough to do a drug itself. Means, as I mentioned, what was our objective to find a lead? Potency. So any structure which gives the highest potency will be called as lead. But are they drug? I don't know. You have to further optimize that and make it into a drug-like property, drug-like candidate. Suppose if you can modify that, then only it can become a lead, right? So that is an example of that is the meaning of lead compound. So I think I'll stop here for this because this we have anyway covered, and the lead optimization we have covered. I think I'll get into the other topic which we need to cover, and that would be uh, computer aided drug designing, right? So if you can see my slide, this is so computer aided drug designing. Basically, just a cartoon to showcase that this is your compound. This is the capsule. Basically, this is the drug, and the designing is happening using computer. Something you are doing there. That's why you get. So this is called as computer aided drug designing. Correct. So, computer-aided drug discovery, basically structure-based design. So now, as I mentioned, there are libraries of structures available. There are libraries of molecular targets available. So based on that, bioinformatics, using that, when you try to look for the biomedical application, so those softwares when you use actually, and then try to come up with a structure which gives you perfect binding to a target, right? So computer aided programs really helps you in doing so. I mean, in nutshell, we are using a computer, so naturally computer aided drug designing is what is really helping you in that, using a computer. So it's called computer aided drug designing. So if even if you are looking for selectivity, now that all the structural information data set is available, you can typically look, whether your structure is selective or not, how much affinity it has to the ligand, to the receptor, right? Are there any metabolic liabilities? If there are, how can you intervene that, right? Are there any potential to form a toxicity in a ligand? Where does in that pocket it really binds? What is the interaction? Is it reversible? It is non-reversible. What kind of a forces are applied, van der Waals force or covalent binding, what kind of, all those things can be studied, not by really synthesizing the molecule, simply using a computer. That's why we call it as computer aided drug designing. Correct. So as I mentioned, there are a lot of platforms available from IBM, Dell, and Dawning, and you imagine more than 6 million chemical data. Imagine this much of chemical base, more than 6 million, right? That much of chemical database is available, correct? You can do all kind of molecular docking which you are looking for. You can identify a particular orientation structure in form of a pharmacophore, and do a typical modeling in which way in 3D structure it should move, where it should bind, how long it should bind. All those things can be done, right? So this was not there a couple of decades ago, but now it is available. So all IBM, Dell, Dawning platforms are available, and the professional softwares, just to name the few, Series 2, Catalyst, Insight 2, all these are the softwares which are available, which can be used to do the drug designing, right? You can even study the structure activity relationship, or if you are looking for a novel drug designing, you can always use this molecular docking, pharmacophore, modeling, all this thing to your advantage in computer-aided drug design. Okay, so drug design with help of computer, as I mentioned, it can be used in different stages, where it helps you first to even hit identification. Using just by virtual screening, we can do the screening of the hit molecules and identify one hit, right? From that hit to lead optimization, we want to increase the affinity, we want to increase the selectivity, or we want to reduce the metabolism. All those things, just by modifying the structures, first you can study using in silico this softwares, if affinity has increased, if your selectivity has increased, right? So from hit to lead optimization, and then further optimization of lead also you can use computer aided drug design. So it has its role from target identification to drug designing all the way up to lead optimization. Any point in time you can use computer aided drug designing as an in silico tool before actually doing anything physically, that is actually synthesizing the compound. You can do all this thing, correct? Even graphic, earlier it was always the graphics was not that much, right? But nowadays gaming softwares are there, and graphic cards are available, so same graphics has come into play even in computer aided drug design. So now you see, this is just an example. This is beautifully, I mean, it's a one way, so I can't move it, but when you are actually using the modeling, you're doing the modeling, you can actually move around 360-degree view you can have for this. So you see the structure of your molecule and the binding under the pocket in the ligand where it is going. You can have a look at it from a 360-degree view, right? So beautiful graphics are nowadays available for this. So this is just an example in a particular pocket, which part of your structure, pharmacophore wise, if you say, is actually getting attached where, in which particular space, what is the orientation, what is the energy, everything can be seen using computer aided drug designing, correct? You see, dynamics can be studied as well, even quantum mechanics. Just I'm just giving you a few examples to just to increase that curiosity among you, so that you can go back and get into details of more details of computer aided drug designing and try to learn something, especially for chemists or bioinformatics scientists, right? So that's the role of computer aided drug designing. Similarly, uh, of course, QSAR is definitely a part of it, but compounds have a biological activity, right? And now using this softwares, when you actually design new compounds with improved biological activity, it could be improved selectivity, it could be improved activity, it could be improved metabolic profile, anything. If you improve the biological property, so that is called QSAR. So one way is you have a compound structure and some activity is known in terms of potency. Now, if you use computer aided drug designing, if you use QSAR modeling, and based on that, if you improve the structure for improved biological activity, that is called QSAR, right? So what is basically QSAR? It's basically a mathematical relationship between a biological activity of a molecular system and its geometric and chemical characteristic. So how it really helps you is, it gives you some mathematical relationship between what? Between the outcome of your molecule, which is a biological activity, right, of whatever molecular system, and in terms of its placement, orientation, like in geometric, and the chemical characteristics. So that is called as quantitative structure activity relationship, QSAR. Okay, so it's given by F, which is basically molecular fragmental properties. That it is revolving around the activity. Okay, so it attempts to find consistent relationship between a biological activity and the molecular properties, right? Because you're getting to the level of a molecular structure at a protein structure, at 3D structure of those ligands or protein structures. So it says that relationship between the biological activity and the molecular properties. So in a way, so that you can come up with some rules which can evaluate the activity of new compounds. So once you know that binding, once you know that relationship of biological activity and the molecular properties, you can come up with some way to screen or to evaluate the activity of new compounds. So that's called QSAR. Okay. So basically, it accounts for the parameters it accounts for is hydrophobicity, as I mentioned, biological and molecular properties. So hydrophobicity of your molecule, the topology, electronic properties, right? Or the electronic in terms of photons, electrons, the static effect, all this thing. And from a statistics point of view, the statistical analysis, basically when you try to determine all this empirically, so you're trying to measure this. So chemical measurements and the biological of a biological assay, that's what basically you try to achieve using QSAR, right? So structure property correlations, when you look anything from a molecular structure, it will have few components, right? One of the important is the intrinsic properties, the molar value, volume, right? The charge distribution on that particular molecule, the molecular weight as such of of that molecule. There'll be a chemical properties attached to it, like a pK, log P, solubility, stability, and there'll be some biological response attached to it. What would be? It would be either an activity, or it could be an adverse effect in terms of toxicity, or it may be a transformation, a biotransformation, either by CYP1 to metabolism. So it will be biotransformation. So finally, pharmacokinetics. So structure property correlation is like any molecular structure will have three different components to it: its own intrinsic properties, chemical properties, and the biological responses it can elicit. Okay, so that is the molecular properties it makes use of. And what are the molecular descriptors? Whenever descriptors you're talking about some numerical values, right? Some values which can characterize the properties of a molecule will be called as molecular descriptors. So they are generally in four different categories: either topology, geometrical, electronic, or 3D descriptors. So topological, when you say, they are derived directly from representation of structure. Means when I talk of any structure, it can be an atom, right? It can be the bonds available on the that. It can be the if there is any substructure attached to that, right? The molecular connectivities, kappa indices. Please don't get confused, but this is more to do with the structure of a molecule. So all the parameters, molecular symmetry, all those topological descriptors are considered under topological descriptors. Geometrical, as you can imagine, is more from a principal moments of inertia, the solvent accessible, mainly the surface area availability, what is the charge distribution of that surface area, and eventually what is the molecular surface area available in a three-dimensional view. That will form a geometrical descriptors. And then electronic. So as you can see, what is electronic? So quadrupole or a dipole. What is the polarability, like polarizability? What is the energy, dielectric energy on each of those bonds? That forms for an electronic descriptors. Molar refractability also. And same way, the fourth class within that is hybrid or 3D descriptors. So again, geometric atom pairs, topological torsions, spatial factors, all these things are included within hybrid or 3D descriptors. So please remember, whenever you're talking of a QSAR, all these factors are taken into consideration to give you the modeling system which is available, right? Topological, geometrical, electronic, and 3D descriptors. So these are all taken into account, and then it is available. So now, even after being so precise, there are still some limits of descriptors. That the data set should contain at least 5 times as many compounds as descriptor in the QSAR. Okay, only then those predictions and those understanding would be correct. If the data set is more than at least 5 times more than it has more than 5 times more compounds, okay, then the QSAR descriptor in a QSAR. So that is the only limitation, but still I would say it is still by far most accepted technique for of doing quantitative structure relationship. So if you really want to understand and the calculation part of all these molecular descriptors, I just put few freely available tools online. You can review that like CDK, Power, Mold, Paddle. All these are freely available on net. You can really click on that and download that, and then further understand how exactly these descriptors are calculated. If you talk of QSAR softwares, so these are all commercially available softwares like Tripos. So you have ComSIA, WallSurf. I think WallSurf is also very common QSAR software. Same way Catalyst, Sirius. So this is also a software available. Then docking softwares are also nowadays available. So computational chemist can actually do this docking process for you and understand the molecular interaction and the binding of your structure with a ligand and all. So Kunz, Langware, MSI Catalyst, these are all the well-known softwares for docking. QSAR I already shared for computer-aided drug designing also, which are the softwares available. So as and when possible, you can actually go for any such descriptor, and online also you can try and understand. But I would say that if you want to get into bioinformatics or you want to get into uh, computational chemistry field, then you should definitely study about QSAR, about docking, and about computer aided drug designing. Okay, so with that, I think I will stop here, and we have in brief talked about computer aided drug design, QSAR, and about the different fields, especially involved in target identification. So I'll stop here, and if there are any questions, I'll answer. If not, then very quickly we will go back and do the some revision on few of the topics which we have earlier. Okay, so I'll take a gap for a minute or two to answer any questions, and then we will jump dive into the revision part of at least one of the topics which we can do, and remaining topics recap we will do it on Monday. So I'll be more than happy to answer any questions if you may have right now. I can understand.
Students, anyone having any queries, please respond in the chat box.
Yeah, I can understand the information is very overwhelming within one class, but take your time. I mean, later on also you can connect with me if you have questions on this. I'll be more than happy to help you through if you have any questions going forward.
Sir, I had put in the chat box one question.
Oh, okay. All right. Okay, so this is the question how how are hepatocytes and enterocytes collected? So fantastic. So yes, you can collect this. Of course, not from human, but from lab species, you can definitely collect, and then you can apply for an IAC approval internally with your institute from a rat or a mice, and we can really do this actually in your lab. And all you need is a permission to use those animals, because as you may understand, these are terminal experiments, so animal needs to be sacrificed basically, because you will have to cut open the animal, and then you have to collect this. So basically, generally when I used to do it, nowadays they are commercially available enterocytes, hepatocytes, so practically you don't have to collect it. But primary cultures we used to collect when I was studying or during initial phase of my career. That time you have to do the perfusion, right? And after perfusion, with the perfusion, you can collect this. primary culture, you can even do the scrappings and all. But as you can imagine, it's a surgical process. So you have to cut open the animal, and then all this entire exercise has to be carried out in a very sterile environment. Otherwise, at least we used to face a lot of issues of contamination. So you can definitely do this, but all you need is an animal. And for animal usage, since it's a terminal experiment, you need a permission. But if you have that in place, just like a primary culture which we isolate, you can do it from an intestine. There's a huge intestine surface area available for you to collect, and for even from liver also we can do the primary hepatocyte cultures. So it is doable. How we know the particular drug for selected target? All right, so this is exactly what we have discussed, right? That once you decide a target, you have to actually validate that target, right? So target, as we have discussed, could be an enzyme, could be a pathway, could be a signaling pathway, could be an enzyme or a receptor. So once you have identified that, then you need to check whether by you should know the functions of that particular molecular target. That okay, this is the receptor. Is it that by blocking that receptor, some the disease state which is come can be prevented, or maybe by releasing certain pathway, can I intervene some function, thereby I can treat the disease? So you don't know initially, so you have to find out this. So how do you do it? Using computer aided drug designing. So these structures are all available, right? You have the molecular target structures also available. Now you look for where those binding can happen, where your structure, your molecule binds perfectly with the ligand, right, or with the receptor. If that binding is perfect, means you are able to block that. So now that will just tell you whether you are able to, suppose, block that receptor or a pathway or not. Whether it will treat disease or not, you need a biological assay in place. So that biological assay can be in vitro or in vivo. So in vivo, you should have a proof of concept. So how do you do it? Nowadays with rats and mice, genetic models are available. So basically you can actually, if it is a genetic target, I can really knock out that particular target. Suppose if I feel that this particular gene, if I do some modification in that, then my disease will be cured. I can remove that gene and do a mutation. I mean to say, sorry, I can remove, of course, by mutation, I will remove that, and then I will breed those animals. After few generations, I will get an animal in which there is no that particular gene available at all. Now I can simply see if that gene itself is not there. If I try to induce the disease, does it happen? If it doesn't happen, means that my gene selection was correct. So now I know that if I target this gene, disease is going to be cured. So this is one way of validating my target. Other ways, you can actually do a disease model. Like a diabetes, it's easy. I don't need to modify it so much genetically. I can simply treat them with streptozotocin, have a increased glucose in mice or rats, and then treat them with my drug and see if glucose level comes down. Means I know it's working, right? But for molecular target validation, one is computer aided drug designing you have to use, and second is for a biological assay, you can do a knockout or knock-in mice or rats you can use, and further validate that the target which you are working on is really apt. I mean, it is really optimal. Okay. How we know the particular? Okay, this is done. My yesterday's question is e-Pathshala free program or payable, sir? How is it helpful in getting good placement? Good. So e-Pathshala, you asked. Of course, it is payable program, but I think since it's a, as I said, it's a government initiative. This is a very nominal, hardly 1,000 to 2,000 rupees would be there for the fees, and it how it helps you in good placement. So good. It's a good question. Of course, it will help you to enhance your knowledge. Okay, because there are e-Pathshala courses now in microbiology we do. So in microbiology, 1 week within 1 week time, what we try to cover? We try to cover sterility testing, preservation testing, food testing. So how it helps? All these are the testing which generally happens in the industry. So if you really are interested in doing a job where this thing can happen, then you can actually go and use this certification and your knowledge, and you can get a breakthrough. Suppose you want to get into research, at least within that 1 week you will understand the concepts of drug discovery development, so it will give you more confidence to face any interview. Okay, same way analytical. So you might have studied the theory of those in this courses of e-Pathshala. Within 1 week we try to tell you the principles is okay, but the application part of each of this instrumentation, say HPLC, mass spec, spectrometry, biological sample handling, how do you do sample processing, what are the guidelines applicable, so that really enhances your knowledge, and it helps you to, although you have required qualification, you might have studied the principles of how these different instrument really work, but you may not be knowing the application part of it. So this will further give you confidence to go and appear for the interview. So e-Pathshala only enhances your knowledge, but those courses are designed in a way that they are highly relevant to the job requirements, what all work is going on in the industry. So that's where this can help you. Okay. Any other question?
Student, okay, sir. Students, anyone having any queries, please respond in the chat box. Regularity affairs department is important of industries while doing a job or not.
Yeah, 1 second. Okay, can you just repeat the question? Regulatory department, you are saying?
Yeah, hi. Put in a chat box, sir.
Okay. Regulatory affairs department is important for industries while doing job or not. Yes, of course, it is important. We have talked about it yesterday in detail. It is very much important actually, because regulatory department is one of the prominent department in most of the companies. They decide whether the target which you have selected, disease target, where the whole project is going to run. Do you have opportunity to file a patent? Or if you selected any target, your regulatory department can tell you that if there is any patent already filed on that target, so you cannot even start your project if you don't have a regulatory department. And regulatory affairs always keeps you updated with the recent advancement or recent changes in the regulatory guidelines. So you will be busy doing your science, but you have to align it to regulatory requirements. Otherwise, you can't submit your data. So that's where it really helps. Docking is useful on formulation development. Okay, so docking is generally, I think docking, as per my this thing, docking generally can be used in enhancing the in our understanding of the molecular structure, okay, of the target and our binding of our structure to that. That's where docking studies really helps in a great deal. I don't think it provides much help in formulation. Sometimes, I mean, sometimes people have used it to their advantage just by using docking uh, data for formulation development. But otherwise, see, whatever formulation efforts you are trying to put in, that is basically to uh, enhance the underlying issues, either solubility, permeability. So basically, it's not going to change the structure of your molecule. Docking can help you in formulation development only when you're trying to change the structure itself. Say, for example, nanoparticles. So you want to see when I do the nanoparticle, or say encapsulation using carbon dot or from the wooden source, when I want to use the encapsulation material or using graphites, I'm going to mask my drug with that. So now the structure has slightly changed. I can use docking there, and then want to see that now does it become a PGP substrate, right? So I have a PGP protein structure available, and I can see does it really bind there, or it doesn't bind. So that way you can use it. But otherwise, I think it is used much earlier than when you reach to the stage of formulation development. Is there any scope for a regulatory affairs department? There is a huge scope. As I already mentioned, each department will have a quality assurance unit and many of the companies, big companies, they do have regulatory affairs department also. What is the best correlation method to perform QSAR studies for nonlinear data? What is the best correlation method to perform QSAR studies? Okay, so I think I personally have not used QSAR, but I can just deep dive into this couple of things, and maybe I'll tell you in the next class. If your data is non-linear, then let me just see what best correlation we can use in QSAR. Okay. So fine. I mean, thanks for the very relevant and very interesting questions. I will be more than happy to discuss further on QSAR for a non-linear data. Okay, great. Any other question, folks?
Students, anyone having any queries, please put in your chat box. Please respond. If you're having queries, please put in your chat box. Otherwise, put in your chat box as no query or no doubts. Please
Okay,
respond.
yeah. So I think we still have time, right, Maheshwari?
Sir, once again.
We still have time, right? About half an hour.
Yeah, half an hour we have.
Okay, so what I'll do is then let me just do a quick recap of one of the topics so that at least we cover up that. Okay, if there are no
Yeah,
more
okay.
questions, let me utilize the time and then take you through the topic where we can do the recap, a quick recap. Okay,
Yeah, okay.
so
You can recap, sir. No
yeah,
one putting a question.
no worries. Are you able to see my screen now? No. Okay,
It
one
is
second.
blur.
No
Your
worries.
video also blurred.
One second. Are you able to see now,
Yes, sir. I
Maheshwari?
can. Yeah, I can. You can continue.
Yeah. All right, so we'll since we have some time, let us do a quick recap of one of the couple of topics which we have covered very initially in first week of our training. So let's talk about aqueous solubility. So as all of you know, I think we have had detailed discussion around solubility. Solubility, maximum amount of chemical which dissolves in a volume of solvent is called solubility, correct? If you do not have data on solubility or understanding, then whatever data which you obtain out of your metabolic stability or reaction phenotyping or CYP induction or CYP inhibition, you will get erroneous data, correct? If your solubility is low, less than 1 microgram per ml, then also whenever you do permeability assays, say PAMPA, say CACO2 assay, say protein binding, while doing all these assays, your solubility would be, your data would be unconclusive. Okay, you may feel that my drug is not permeable, but in reality the drug is not soluble, so it is degraded and you're not able to detect that solution. So of course it is permeable, but since it was not soluble at all, it did not ever cross the barrier, so your interpretation could be wrong. So that's why you should understand about solubility, correct? And you can also concentrate on this particular slide on solubility, which is called BCS class of classification. So by and large, the compounds which are under research or in the phases of drug discovery development can be classified as BCS classification of solubility based on solubility, class 1, 2, 3 and 4. Class 1 is the best compounds, so if they are highly soluble, highly permeable. Class 2, low solubility, high permeability. Class 3, high solubility and low permeability. And class 4, low solubility and low permeability. We wish that our compounds fall in any of the classes of 1 to 3, but unfortunately most of the compounds fall in class 4, which is low solubility and low permeability. Correct? So the solubility is definitely low, but your permeability is also low. But now with formulation development, as somebody has asked a question on formulation development, now we have tools and techniques available where you can actually modify, even if your compound is in class 2, 3, 4, we can really make it look like a class 1 compound. How? Say compound is having low solubility. What you can do is reduction in the particle size, right? Make that compound into a salt, add some surfactants, use nanoparticles, or use cyclodextrin complexes, do some pH adjustment. By doing all this thing, you can make low soluble compound into a high soluble compound. Okay, if your compound is having high solubility but low permeability, then you can increase permeability by adding an absorption enhancing excipients, right? If they are substrate to the efflux transporters, then we can add efflux transporter inhibitors, correct? You can put it into a lipid filled capsules, right? So all these things can be done in order to increase the permeability. So a class 3 can also be made into class 1 compound. Suppose class 4 compound, they don't have solubility, neither they have good permeability, then you can actually first an easy thing is to do convert it into a prodrug, as we have discussed about prodrug. Now prodrug can have enhanced solubility and enhanced permeability, or you can add co-solvents, or you can use encapsulation, or nanoparticles, liposomal deliveries, lipid filled capsules, salt forms. So these are all the means and ways by even class 4 compound can be converted into class 1 compound. So eventually, if they have got good solubility, good permeability, overall viability will be good. So kinds of solubility assays are being done initially. One is kinetic, one is thermodynamic. As thermodynamic suggests, it is based on the equilibration over a period of time. Kinetic is pretty fast, quick in a high throughput fashion. Generally, in a research environment, almost all the molecules will have solubility issues. We quickly dissolve them in a DMSO, prepare a stock solution, and then in a well plate, in a 96 well plate, we incubate it. Okay, and then we just simply do the shaking, mixing, take the supernatant, inject it, and quantify. Whereas in thermodynamic, you start with a solid, long mixing till the equilibration is arranged. And since it is a long mixing and you keep on adding till maximum equilibration is maintained, it is not high throughput either. So by kinetic solubility, you can do, since well plate format, up to 150 compounds a day, whereas in thermodynamic, hardly 20 compounds a day. Okay. I don't want to get into details of methodology, but it can be done in your lab itself. At least the equilibration one, the thermodynamic can be done easily. Different glass containers you can take, and then add your compound to that desired volume of water. Keep it up for mixing. After 24 hours, take the sample. After 48 hours, after 72 hours, look for the solubility. I mean, look for the quantification of the compound. When the concentrations measured at least in two last vessels do not differ by 15%, that is the accurate solubility, right? Then we talked of octanol solubility. So octanol solubility, again, why? Because it's an organic compound, it's a fatty oil. The structure, it's a lipid, so structure mimics to that of bilayer lipid bilayer in intestinal membrane. So we hypothetically feel that it mimics that structure. So it's also called as log P, right? And it is denoted by letter P, which is nothing but concentration of your substrate substance in organic versus in aqueous. Or you can find a partition coefficient, also known as log P, also known as octanol solubility. Very easy assay to do. Mix it up with water, put it for shaking, add amount, I mean, take the concentration of your water or concentration of your compound in a water, add to that same amount of octanol, mix it up, shaking, and then centrifuge and take octanol layer and take water layer and look for the concentration in each of the layer and find out the partition coefficient. Plasma stability assay, we have discussed in detail. Any molecules, if they are liable to degradation in presence of hydrolysis or esterases, then plasma stability should be one of the initial in vitro assay to be done in your lab. Okay, so enzymes, cholinesterase, aldolase, lipase, dihydropeptidase, all are present in blood. So if your molecule is having liability or degradation potential from any of these enzymes, then plasma stability should be done very initially. So these are most common functional groups, which if you see this in your structure, then you can think of having plasma stability assay very early in your drug discovery. Means if your structure is having ester, amide, carbamate, lactam, lactone, sulfonamide, then your structure could be prone to plasma degradation. So, for prodrugs, of course, because as we have discussed, why do we prepare prodrug? Because it may have some issues with solubility or permeability, or both the issues. So, to counteract that, you prepare a prodrug. Now, after prodrug, it gets absorbed, but once it comes in the circulation, you want an active drug, right? So, prodrug should convert into active drug in presence of enzymes in plasma, maybe hydrolysis, maybe esterases, maybe dipeptidyl peptidases, lipases, anything. So you would like to test that thing initially rather than dosing the prodrug to animal and then looking for the active drug in blood. You can do it in vitro assay and see that whether your prodrug is actually releasing the drug in presence of blood, in presence of plasma enzymes. So for prodrugs, it's one of the very important assay to be done very initially in drug discovery, right? So importance we already studied. Strategies to improve, I already talked about this structure. So a chemist, if you go back to chemist and say that your structures, is there any possibility that we can modify these structures and still the potency will remain same? If so, please do so, so that plasma stability can be ensured. So a chemist can help you in that, right? So again, very easy assay to do in your lab environment. You can very well do this experiment. Generally, it can directly be done in plasma, but sometimes they'll have a lot of background if you use directly the plasma. So people use it along with 80% phosphate buffer. Okay, at 7.4 pH. So add your drug to it and then incubate it at different time point. Take out the sample, denature the protein, do the sample processing using protein precipitation, liquid extraction, or solid phase extraction, and the sample final you can inject into HPLC or a mass spec, quantify your drug, and compare it again different time points. So you will come to know if your drug is stable in plasma or not, and what is the percentage of instability. Okay. Then we talked of accumulation, gastric fluid, and intestinal fluid stability. Very important because the first organ which drug will see after oral dosing would be stomach. So it is exposed to acidic pH, then it goes to intestine where there will be a basic pH or neutral pH. So you would like to see the effect of drug in vitro. What is going to happen in an acidic or a basic or a neutral pH condition in intestine. Again, very easy to do. You can use simulated gastric fluid. You can prepare in your lab. Only additional, you can use a sodium chloride as usual, and a pH can be adjusted to 1.2, and you can add pepsine to give a similar environment as it is available in stomach. Whereas when you go to intestine, you can add, instead of pepsine, you can add pancreatine. So it will mimic the intestinal fluid and check the stability at different time points, and you will come to know whether your drug is stable in gastric and intestinal fluid or not. So very important assay to do very initially. Then we talked about microsomal stability assay preparation part also. So again, subcellular fractions, what are microsomes? They are proteins. Subcellular fractions mainly derived from endoplasmic reticulum. Why they are important? Because they do express cytochrome P450 enzymes, main phase 1 enzymes. So you can make microsomes in your lab very easily, right? So all you need to do is initial animal procurement, or you have to have a permission from ISC, use a isolate the liver, isolate the microsomes, and actually do the microsomal assay. So basically, you have to sacrifice the animal, cut open the animal, infuse the liver with ice cold homogenizing buffer, then take out that pale liver cut it into pieces, and then take small pieces of 4 grams each. And then either using a homogenizer, you can further cut it into smaller pieces, or using a mortar pestle, 10 strokes. Not very vigorous mincing, but very gently you can do it. And then centrifugate that for 12,500 g. Then discard the supernatant, whatever pellet you have got. Again, resuscitate in a buffer, homogenizing buffer. And now you have to 1 lakh G, a ground mounted centrifuge, temperature controlled centrifuge at 4 degrees you have to do for 70 minutes. Again, you have to repeat the process for G at 45 minutes, and then you have to resuspend the pellet what you got into the again microsome buffer, right? And your microsomes are ready. Before you actually can use, you should have the protein concentration. So generally go by biochemical methods, any biochemical method to estimate the protein. We generally have used always Hartree-Lowry method, or the identification of proteins. And then store it in -70 to -80 deep freezer, and microsomes are ready. So from microsomes, when you want to do an assay, take out, thaw the thing, use required protein concentration, that much of microsomes, add your substrate, which is your drug, add MgCl2, and a cofactor which will start the reaction is NADPH, and then your reaction starts at different time points. Incubation, you can take out the sample and look if it is metabolically stable or not. If it is unstable, over a period of time, how much metabolism is happening, you can easily identify. Each time when you take out the sample, to stop the reaction, you have to add ice cold. I will re-emphasize, please use ice cold methanol or acetonitrile. And whatever data you get from that, we can calculate intrinsic clearance, right? So elimination rate constant, half-life, and volume. This three things actually comes into the play when you calculate the intrinsic clearance. So that is about microsomal stability. Then we have also discussed about absorption. So as you all know, absorption happens from intestine, from the gut wall. There are PGP substrates which are sometimes PGP transporters, which could be efflux or uptake transporters based on affinity of your substrate or a compound to those transporters. Either it will efflux the drug back into the intestine, or it will permeate that. So that's why absorption is one of the rate limiting step for bioavailability. Okay, so from this diagram you can see once it gets absorbed, either with interaction or without interaction with the transporter protein, it gets absorbed through portal vein, it goes to the liver. Even in intestine, we already have discussed this even yesterday, that enterocyte can really do the metabolism. It can be excreted here, or through portal vein it can go to liver and it can get metabolized there, and metabolites are more polar, so it will be excreted out of your body through renal mechanism, or it will go into the systemic circulation and it goes to different organs and even to the site of action. So that is the absorption followed by the bioavailability. So this phase which is happening in the intestinal barrier is called absorption, and whatever metabolism happens in liver and the drug is lost there, that is called liver phosphorylated, right? So this is about absorption and bioavailability, correct? Then let's move on to cell membrane structure. We have seen, I already mentioned lipid bilayer, phospholipid bilayer membrane. So both are lipid bilayers. These are membrane proteins available. The regions, these are hydrophilic regions, and the inner side is hydrophobic regions. Okay. So if you take a microvilla from an intestine, if you zoom this, you will see the structure, the the cellular structure. So this is the apical side, basolateral side. So diffusion can happen transcellular, which is between the cells, paracellular, between the cells, through the cell is transcellular, majority of the compounds, and there are transmembrane uh, uh, mediated, carrier mediated protein, transmembrane protein mediated transport, that is carrier mediated. So these are all the mechanisms of intestinal drug transport. Okay. So again, talking about diffusion, as you all know, can be passive or facilitated. So water and water soluble substances and small lipids, they always go from in form of concentration gradient from high concentration to low concentration, right? So that is passive diffusion. Facilitated, they actually are using this transmembrane proteins to get transported. So that is facilitated diffusion. Then active transport, here what happens is you need an energy in form of an ATP in order to transport the drug or nutrients across the cell. So that is active transport. And these are just to name the few transporters like PEP-T, OATP. These are transport proteins, either efflux or uptake proteins. Based on that, either it can transport the drug the substrate or nutrient across the cell, or it can deliver back if it is a efflux transporter. Okay. So passive diffusion can be transcellular, as I mentioned, through the cell, can be paracellular, right between the cells, and it takes into account the log P molecular weight and polar surface area availability that will define the passive diffusion. Carrier mediated, you can have facilitated transport where through membrane proteins it will happen, or in form of in presence of energy in form of ATP against the concentration gradient, that is active transport. Okay, so this is just how an what do you call active transport looks like, and uh, how a passive transport looks like, and how an active transport looks like, because it is against the energy. And it is passive transport is from always low concentration to high concentration, so this is how it looks like. Yeah. So, what are the factors which can influence? We have discussed in detail. I won't spend much time, but yes, polar surface area, blood flow to the particular organ, gastric emptying time, your affinity of substrate to the drug transporters, either efflux or uptake transporters, and presence of food in the intestine. So all these are the physiological factors. Then there are physicochemical properties like partition theories, ionization state of the substrate, particle size, the complexation, all that can influence the drug absorption from intestine. Then permeability models we have discussed in detail. So there could be a biological model, a cell-based model, or a non-cell-based model, a non-biological system, right? So many years ago, even now, sometimes using the chambers where you actually keep the tissue, and then you can look for the diffusion of your drug back and forth. CACO2 canine adenocarcinoma cell lines. Here you can have a monolayer of cells grown, and then apical and a basal layer you have. You add the drug and then look for whether it is able to diffuse across. After incubation, you can take samples from donor and acceptor and then look for A to B or B to A transport. Then there are non-cell based artificial membrane, like this is IEM, immobilized artificial membrane columns, which mimics the structure of phospholipid bilayer. So this can also give you an idea about permeability. Then there is something called as PEMPA assay, parallel artificial membrane permeability assay. There again, instead of cell monolayer, we use a microplate with lipid and solvents, lipids, because it will mimic the intestinal structure. And then you look for A to B diffusion. Since there are no transporters involved here, no need to see B to A, but we always look for one-way flow from this. And in fact, from these studies, there is no expression of PGP, so you don't know the role of PGP substrate into this. Then there are SPR biosensors also available nowadays with hydrogel, with liposomes can be used, and you can look for the light scattering. And then there are in silico models, as I have talked about. CADD, when you do, there are many softwares available which can take into account the permeability potential of your drug as well, based on the structure. So each of these methods have some pros and cons. We won't get into much of a detail, but very quickly, in excised tissue, we just look for the disappearance from the donor and appearance into the acceptor. So what is the downside? So there is a variability, because the tissue, what you have cut, the size of that, the location from where, the site from where you're taken, all that brings about variability in the data. Okay, and it's a very low throughput because we have to keep it for a long time for equilibration. So that's why excised tissues, this is the downside. For cell based, all CACO2, MDCK made in there be canine kidney cell line, ST29, all this thing can be used, can be automated. Okay, and a beautiful mono layers are grown on a filter support in a multiple well format, generally in 24 well plate format, and it can be measured for the movement of substrate from apical to basal or basal to apical layer, as I mentioned, A to B or B to A. So we can definitely, if you want to understand the role of PGP substrate, just add a PGP inhibitor, verapamil example, generally it is used, and now you see the movement. If it is enhanced, means your substrate is inhibitor, is a your drug is a substrate of PGP, because now that you added PGP inhibitor, you are seeing more permeability, means your substrate, your drug is a substrate to PGP transporter. So to identify that, you can do it in the presence of a PGP inhibitor, verapamil. So this is what extraction ratio, generally apparent permeability, that is from B to A divided by A to B, the concentration that will give you, and permeability, apparent permeability extraction ratio. There are some downsides, because there could be variable expressions of P glycoprotein, right? So whether your compound has any liabilities for that transporter protein, sometimes you can't make out because the expression would be different in different cell lines. Okay, and there could be variability in inter and intraday, the person who is doing the experiment and all, and it's a long experiment because it requires 21 days of culturing. So that those are the downsides of using cell based. In artificial membrane, PEMPA and IAM, these are quick, faster, and easy to do. No, and it can be done in automation fashion, right? Downside, there is no expression of PGP substrate. So any liabilities related to that cannot be predicted from this, or cannot be studied from using artificial membrane models, right? And since they are not live cells, the predictability is also not that great using artificial membrane. This is typically how it looks like. You can put it an apical and a basal layer, and there is a liquid solvent, generally lipid here, and this is the formula how you calculate the apparent permeability from a PEMPA assay. Okay, for the composition of that layer which is being used, earlier phospholipids, hexadecane, unsaturated hydrocarbons, all of them have been used as a layer instead of cell layer in PEMPA assays. Okay, so this is how this is one of the important aspect I have mentioned, that whenever you do permeability assays, please calculate the recovery. Okay, recovery means if I have added X amount, how much I am able to recover either from a donor from or from acceptor. It should be 100%, close to 100%, because whatever, suppose I used X concentration and I am find a recovery only 80%, means 20% is lost. That means percent is some binding is happening either in a well plate, profiling tubes, or it is getting attached to micro plates. So that is a non-specific binding. So for an accurate determination of permeability, always consider calculating the recovery of your compound in that assay. Okay, in silico, they make use of molecular weight, log P, hydrogen bond donor, hydrogen bond acceptor, and solubility, electrotopological state, polar molecular surface area. All the descriptors which we have talked about today, they are used in silico methods. Okay, generally Lipinski's rule of 5 for molecular weight, log P, hydrogen bond donor, acceptor, these are taken into consideration in deciding the permeability using softwares. The downside is they are basically predictions, right? They're not, of course, they are. We try to consider all things, but still they're not actually a real physiological process, right? So sometimes the predictions are completely off. So you still have to practically do the experiment and understand if there are any issues related to permeability or not. Okay, so overall, in summary, permeability can be passive, can be active. Under passive, it can be paracellular, between the cells, can be transcellular, through the cells. You can study this using PEMPA or IAM. You can study this using a cells. If it's an active transport, you're looking for an uptake, then you use a CACO2. An active secretion, you use an MDCK and other cell lines. So this should be the strategy to study the permeability characteristics of a new chemical compound under research. Then we quickly talked about, I think there's only 5 minutes. Let me see just a couple of slides and then we'll conclude today's lecture. But pharmacokinetic parameters we have talked about in detail. Their core, their relationship, how they are interrelated, how do we really interpret this, the preclinical data. And as you know, more than 40% of the cases, there used to be a time when drug fails because of poor PK properties. It's an old slide. So nowadays, it's only less than 10% chances that drug can fail because of poor PK properties. Okay. Pharmacokinetics, we have understood what body does to the drug. That is, drug is going to be absorbed, metabolized, excreted, distributed in the body. So ADME is called pharmacokinetics. What pharmacological action is elicited in our body is by the drug is called pharmacodynamics, right? So the fate of the drug after systemic circulation, either it can be distributed to tissues, or it can go to liver, get metabolized, or in intestine get metabolized and cleared, or it can be excreted via excretion. So excretion plus metabolism, that's why is together constitute for the clearance of any compound. When it gets distributed to tissues, that is the distribution. The parameter is called volume of distribution. So if somebody asks you what are the primary PK parameters, clearance and the volume of distribution, what are the secondary PK parameters, half-life and the bioavailability. Why? Because whatever actual metabolism and excretion is happening, that is defining the clearance, right? The whatever distribution is happening is defining the VD. That's why we call them primary. And from the half-life and bioavailability are the derived parameters, right? Practically, we try to derive these parameters. That's why we call them secondary PK parameters. Okay, so most important parameters for you to understand: clearance, volume of distribution, half-life, bioavailability, and protein binding. Okay, so these are the most important parameters which all of you should understand. So I think I'll stop here, and maybe on Monday when we have a class, I'll try and see if I can take you through a virtual tour. I mean, practically taking my, or I can shoot this and show it to you. Let me just see, between today and Monday, I will decide this, and I'll actually show you a lab. How actually, of course, all of you are from college and all, you would have seen your labs, but this particular facility I want to show you, which is a CPCSA approved facility, and as per the guidelines. So starting from the flooring to the room designing to the operations and the different demarcations within the, everything is as per the guidelines. So I'll walk you through how it looks like. Okay, and we will see a live animal facility. Okay, and we'll do some revisions also if possible, and then would like to wish you all very best of luck on Monday, so that then Tuesday you are ready to appear for a assessment. Okay, don't worry, don't take much of attention like your college exams and all. It will be pretty straightforward, and I'm sure if you have attended all these lectures, all of you, practically all of you, would be able to clear that online assessment. Okay, so I will discuss with Maheshwari how do we do that, maybe through a Google form or something, and you should be able to all clear that out. Okay, so with
So
that,
by Google form only we will conduct the exam, sir.
sure,
Yes, sir.
definitely do a Google form. That's fantastic.
Yeah.
So with that, I just stop here and wish you all very what do you call, a great day ahead, and we'll be happy to answer maybe one or two questions, and then we'll conclude. Okay, one question I could see: why do we prefer analysis pharmaceutical departments? I think I answered this question yesterday. So both are different. Analytical departments, formulation development departments are separate, and regulatory affairs department are separate. Yes, sometimes small companies can't afford to have a separate regulatory affair, so they try to allocate this job to quality assurance department. But big companies, they always have a regulatory affairs as a separate department. Okay? But analytical and formulation is always a separate department. Maybe a very small company where they can't really afford to have demarcation of departments, they might have merged. I mean to say, they might have requested people to do multitasking and do a regulatory affairs job. Even though if they're analytical chemist or a formulation scientist, that's a different scenario. But ideally speaking, they're always separate. Okay. So with that, let me just end here. Thank you and have a great weekend to all of you. And I hope things have become clear in your mind about drug discovery and development, and we have covered in much detail the entire overview of drug discovery and development. So looking forward to meet with all of you on Monday, and let's have some discussion. Some 10 to 15 minutes of time we will have for I think Monday I'll keep more for interaction. So we will have a virtual tour of a lab. Okay. Secondly, we will have about the career path. We will have some questions. I mean, some interaction. I would like to share some role of different fields like chemistry, microbiology. All of you have already witnessed during this 15 days of class, but specifically we'll talk about pharmacology, biotechnology, microbiology, and other fields for their career path growth. And then we'll, time permits, we will talk about a few of the recap of the topics, and that's the plan for Monday. Okay. So with that, thank you for your time, and looking forward to see you all on Monday. Thank you. Namaste.
Thank you, sir. Thank you so much. Students, we'll have again on Monday, tomorrow and on Sunday we'll have a holiday. On Monday at 11 o'clock we'll meet again for the section. And today feedback form is not available because of some technical issues. We'll consider today attendance for all of the students. Don't worry about that. On Tuesday we'll have an assessment about drug discovery development. Okay, all of you prepare. Don't miss the time, and thank you. Thank you so much. The section was closed.